Hello and welcome. My name is Colby Evans. I am a dermatologist in Austin, Texas in the United States, and the past board chair of the board of directors of the National Psoriasis Foundation. I'm here to speak to you today on biosimilars. We'll talk a bit about the history of biosimilars and how they are made, how they are approved, and then about some of the hopes and so far unrealized hopes of cost savings from these medications. They certainly are becoming an increasingly important part of the landscape of biological treatment across the world. These are my disclosures and potential conflicts of interest. You can see these companies that I speak and consult for. And efficacy. One thing about biosimilars that is sometimes new or difficult to understand a little bit is extrapolation. So if you have a reference product, the green box on the upper left of this slide, and you want multiple indications for your drug, as many biologics have, then you have to do multiple independent clinical studies for each indication. You do one for psoriasis, you do a separate one for psoriatic arthritis, you do a separate one for Crohn's disease. When you create a biosimilar, you do analytics in the laboratory to establish a high degree of similarity and biosimilarity, and then you can do a smaller number of functional studies to prove this. So you might start with pharmacodynamics and laboratory studies to confirm biosimilarity, and then you run a phase 3 trial on one indication. So for example, a TNF inhibitor like infliximab might be approved for multiple indications, including inflammatory bowel disease and psoriasis and psoriatic arthritis. And if you did a trial, say just in rheumatoid arthritis, and you showed that it was clinically equivalent, then both the FDA and the EMA will allow you to extrapolate to say that you can then have all the indications for the reference drug while you only did studies in one of the indications. If you want to achieve extrapolation, then you will need to show clinical equivalence to the reference drug. So here we are looking in psoriasis at PASI improvement from baseline, and this solid bar in the center represents the exact same as the reference drug. You then set goalposts, in this case at -15, so inferior to the reference drug, and at plus 15%, so superior to the reference drug. And then your 95% confidence interval of your medication must fall between these two goalposts entirely to be considered clinically equivalent. So this first example is not equivalent because it falls slightly outside the 15% inferior goalpost that we set up at the beginning of the trial. So despite most of the bar being in the equivalent territory, this drug would be considered not equivalent. Similarly, and perhaps confusingly, this drug is superior to the reference drug. So most of its 95% confidence interval falls outside of the 15% superior goalpost on the better side, but nonetheless, this drug would not be considered equivalent. And then these last two, because their 95% confidence interval is fully within the goalposts that we established, they are considered to be equivalent medications that could go on and be extrapolated to other indications. One of the things that we think about a lot with generic drugs is interchangeability and substitution, and you'll find that the world is a little bit all over the place on this for biosimilars. So if you wrote a prescription for a branded reference drug biologic, the patient went to the pharmacy, would the pharmacist have the ability to substitute, say, a biosimilar of the same drug, and would they have to tell you that they did that, or would they just go ahead and do it? Certainly, we're used to this. If you prescribe amlodipine for high blood pressure and the patient goes to the pharmacy, they may well get the generic, at least in the United States, without any communication with the physician. The pharmacist is authorized to substitute it. And so it's long been a question of whether we should have a similar system for biosimilars. In the US, the FDA may approve a product as interchangeable, and then the individual states have pharmacy level substitution control. And so 25 states have passed legislation addressing this, and in some of them they do allow substitution of biosimilars. In Europe, the EMA, unlike the FDA, does not declare interchangeability, and that resides with the member states. And pharmacy level substitution, at least when this slide was written, it was not widely practiced in the EU. In Canada, Health Canada does not designate biosimilars as interchangeable and therefore does not support automatic substitution. And in Australia, they can be designated biosimilars as interchangeable, and the payer bodies get the determination at the pharmacy level, and substitution is recommended as the default policy. So really a lot of differences between different countries as to how interchangeable biologics are, and I certainly expect we'll see this continue to change over the years as biosimilars become more effective and available. So what about actual utilization in the real world? We know that it is quite variable both between countries and between drugs, and I think this slide demonstrates that very nicely. So on the upper left, we're looking at European countries, TNF inhibitors, rituximab, and we can see there has been quite high uptake of those drugs across many European countries, both in the first and second bars of this graph. When you look to the right, some other drugs like insulin and adalimumab, there has been less uptake across Europe than with the early TNF inhibitors, infliximab and etanercept. If you look at the bottom section of this graph, in Canada, Japan, and the United States, and this data is from last year, by the way, there has been substantially less uptake of these biosimilars, and we'll see on the last slide correspondingly less savings associated with that than there have been in Europe. One of the surprises of the biosimilar era has been less than anticipated cost savings. I know in the United States, before these drugs were approved, when we were first talking about them, there were really lofty goals for the amount of savings that would be realized from biosimilars, and that unfortunately has not happened as dramatically as we might have hoped. These examples are from the United States on the left and Canada on the right. So in the United States, if you look at the second quarter of 2017 at TNF alpha, specifically infliximab, uptake of biosimilars in the marketplace, you see it was only about 0.1%. And then almost 2 years later, in the first quarter of 2019, it had only risen to 0.9%. Obviously, we are not going to see the promised savings if 99% of usage is still the reference drug. And at least as of last year with infliximab, that was the case in the US. On the right, we look at Canada first at 2018, so historical data, comparing the OECD predicted savings, both in their moderate and their high estimate, to the actual usage of biosimilars in Canada. So if we look at the moderate estimate in 2018 at the graph on the bottom of this slide, the actual savings were about 94 million Canadian dollars, whereas the potential savings under the moderate OECD estimate was 345.6 million Canadian dollars. So obviously we have saved only a portion of what was predicted. If we look at the forecasted savings in 2021, it is perhaps even worse. We have saved more money now, but of course the estimates have gone up as more biosimilars have become available. So we are seeing still less than half of the potential savings have been realized. So to conclude, biosimilars are an incredibly important and increasing part of the world biologic market. They require a different method of construction and approval compared to reference drugs. Once they have been shown to be clinically equivalent, it is possible to achieve extrapolation and get many more indications than you have actually studied for the drug. And the usage of them, although it is high in some parts of the world, especially Europe, has not yet penetrated to the level that we are seeing the dramatic and promised savings. I certainly think a lot of this is going to change over the upcoming years. We are going to see more and more biosimilars available. Probably we will see higher and higher penetration, although I do not know when we will really achieve the promised savings. That may still be some time yet. Thank you.